Hi everybody, welcome back to our Vidhyalakshmi YouTube channel. Today we are going to see a topic about the blood grouping and RS factor from grade 10. Let us start the video. So the blood grouping, concept is developed by the Landsteiner in the year of 1900. He identified the blood groups A, B, O and ABO. Okay. Then AB blood group recognized by the De Castello and Steny, in the year of 1902. Okay, the human blood contains the agglutinogens, it is called as antigens. Antigens and agglutinins are called as antibodies. Both will be present in the blood group. Okay, then antigen found on the membrane of the surface of RBCs, the antigens. Okay, antigens, I said agglutinogens, it is also called as antigen. It is found. Still there? Can you able to see? Yes. Hi everybody, welcome back to our Vidhyalakshmi YouTube channel. Today we are going to see a topic about the blood grouping and RS factor from grade 10. Let us start the video. So the blood grouping, concept is developed by the Landsteiner in the year of 1900. He identified the blood groups A, B, O and ABO. Okay. Then AB blood group recognized by the De Castello and Steny, in the year of 1902. Okay, the human blood contains the agglutinogens, it is called as antigens. Antigens and agglutinins are called as antibodies. Both will be present in the blood group. Okay, then antigen found on the membrane of the surface of RBCs, the antigens. Okay, antigens, I said agglutinogens, it is also called as antigen. It is found on the membrane of the surface of RBCs and antibodies will be present in the plasma. The blood groups, how we can classify the blood group? It is of 4 types: A, B, AB and O. These are the common blood groups which can found in the humans. So an individual has any one of this blood groups, that is AB blood group individual, and A blood group individual has antigen A. The blood group A has antigen A over the surface of RBCs and antibody, that is anti-B, present in the plasma. In case of B blood group individuals, the antigen B is present on the surface of the RBCs and antibody A is present over the plasma. Okay, antigen indicates a blood group and antibody is opponent blood group. Okay, then AB group individual has both the antigens. So the A and B both will be present over the surface of the RBCs and the antibodies is totally absent. Okay, then O group individual, so they do not have any antibodies, either R nor B. Both antigens will be absent in the RBCs of the blood group, and then antibodies A and B will be present in the plasma. Okay, so A group has antigen A and antibody B, and B group individual has antigen B and anti-A antibodies, and AB blood group individuals has anti-A and B both on the surface of the RBCs and antibodies are absent in the plasma. In case of O blood group, antigen A and B both will be absent on the surface, but the antibodies related to this will be present. So we can see this tabular column, how the antigens and antibodies will be present in the different kinds of blood groups. The blood group A, they have the antigen A. Okay, antigen A indicates a blood group of this individual. Okay, and the plasma of this A group individual has anti-B, and they can donate only to A blood group and AB blood group. The receiver, they can receive the blood from A group as well as the O group. Here anti-B, sorry, this is blood group B. So antigen, the presence of antigen is B over here. The RBCs contains antigen B and antibody here is anti-A. So which is against to anti-A. Okay, then they can donate the blood to the B and AB blood group. Okay, they can donate to the blood to the group individual which has B group as well as AB. Okay, they can receive the blood from the B and O. They can receive the blood from the B and O Then AB blood group individual has the antigen A and B. AB blood group individual has anti-A and B. So the antibodies in the plasma is totally absent. They can donate the blood to the AB individual, and then they can receive the blood from A, B, AB and O. So that we can call them as a universal recipient. We can call them as a universal recipient. AB blood group is called as universal recipient. And then O blood group individuals, they do not possess any kind of antigen over here. They have both the anti-B and anti-A antibodies. They can donate to the A blood group, B, AB and O. So they can donate their blood, all the blood groups, so that we can call them as a universal donor. So only they can receive the blood from O group individual alone. Okay. The next thing is, we can see how the antibodies and antigens will be present over the blood cell. You can see the blood cell RBCs. You can see anti-A present over the surface of RBCs, and B blood group, they have different kind of antigens over there over the surface of the RBCs. And group AB, both the antigen A and B will be there, and group O, they do not possess any kind of antigens. Then antibodies in the plasma, A group individual has the anti-B antibodies, and B group individual has anti-A antibodies, and group AB has no antibodies, and group O, they possesses anti-A and anti-B antibodies. So antigens in the red blood cells, so antigen A blood group has the A antigen, B blood group individual has B antigen, and AB blood group, they possesses A and B antigen, and group O, they do not possess any antigens. So you can see how we can diagnose the blood group in the laboratory. Okay. So they will take a sample of blood and they will add the antigen anti-A and anti-B, where they get the agglutination. So they belongs to that kind of blood group, type A, that is agglutinogen A agglutinates with the anti-A. You can see. Okay, where we can get the agglutination, the person belongs to that kind of blood group. So blood donation, it is also called as blood transfusion. So before we go for blood transfusion, we must check the donor as well as the recipient blood group, because there should be a matching between them. Okay, then only they can donate the blood. So when the blood group mismatched, if the person who has A blood group, if they receive the blood from the B, what will happen? So they leads to the agglutination. They leads to agglutination, that is clumping of the antigen. Say for example, blood group A person will have the anti-B antibodies. So if they get blood group from the B, blood group A person has received blood group from the B, there will be a anti-B will be present in the plasma of the A person. So there will be a agglutination, that is clumping of this both antigen and antibody will get clumping. So that may lead to the death over here. Okay, that is the reason why we should not give the mismatching. Okay, so we should go for the previously we have to go for the different kinds of testing. After that only we can go for blood donation. Person with AB blood group, they are universal recipient, in the sense they can get the blood from the A, B, AB and O. So they can get these kind of blood groups. So they are called as universal recipient. The person who has O group, they are universal donors. Donors in the sense they can give blood, donate their blood to all the kind of blood groups.